Hello students, welcome back to your botany class, and today I would like to start a new chapter, chapter 11 of your textbook, Biotechnology, Principles and Processes. So we'll study only the principles and process in this chapter, while the application of biotechnology will be discussed in the next chapter again. Okay, you are familiar with the term biotechnology, and as it is an integration of biology and technology. And biotechnology is concerned with the exploitation of the biological organisms. It can be the microbes, it can be the plants, or animals. The components, the exploitation of these organisms, their components. It can be the any of the cellular organelles, or it can be even with the nucleus or the chromosomes or the DNA. So, exploitation of the microbes or microorganisms to produce substances, for the welfare or for the human use. And biotechnology can be defined as the application of the principles of science and technology, to the processing of materials, or biological agents for goods and services. So it is concerned with the application of the principles of science and technology or engineering principles to the processing of materials by biological agents for goods and services. So biotechnology can be, in short, it can be described in that way. Okay, so biotechnology developed in two phases. And the first is the traditional, and the second is the modern. So the first phase of development is also known as the classical biotechnology or traditional biotechnology or traditional phase. It's concerned with the production of beer, for example, this alcohol, then beer, wine, vinegar. So all these production are example of traditional or classical biotechnology, and it was practiced by our ancestors way back. And of course, all this production of these substances or products are still very important at the industrial level. And the traditional biotechnology, it is concerned with the natural capabilities of microorganisms, natural capabilities of microbes and of plant cells and animal cells. While this modern biotechnology, it's more advanced than the traditional. And so it is concerned with the exploitation of the genetic material. It's concerned with the exploitation of the genetic material, or concerned with the exploitation of the DNA. So in the modern biotechnology, the genetic material or the DNA, as we have already discussed about the structure, the molecular components, how it replicates. So this genetic material is exploited, it's manipulated, it is cut, it is isolated from the nucleus in case of eukaryotic cells, or it is isolated from the nuclear region of these prokaryotic cells. They are cut into fragments, and then they are again inserted into some organisms, and then those organisms can produce or can even make multiple copies of this DNA fragment. So this modern biotechnology is concerned with the manipulation of the genetic material to produce substances or to produce multiple copies of the fragment of the DNA that is inserted. Okay, so as we have discussed that in sexual reproduction, during the process of sexual reproduction, variation arises due to the exchange of chromosome segments. Variation arises, and many of these variations are useful and they can be exploited for further use. But variation does not arise during asexual reproduction. It is absent, or we do not see variation in asexual reproduction. So during hybridization or plant breeding, both the desired and undesired genes, they multiply in the hybrid. While in this recombinant DNA technology, we see the variation. Since it is concerned with this manipulation of the genetic material, which alternately it's also known as recombinant DNA technology. So since it involves the application of the engineering principles, and to engineer means to construct, to plan, to construct, to build, and manipulate. So to manipulate a substance or any macromolecule towards a desired or predetermined end. So this modern biotechnology or the recombinant DNA technology, it involves the manipulation of the genetic material towards a desired or predetermined end. Okay, so as we have said, this DNA, for example, a particular fragment of DNA isolated from the nucleus or from the nuclear region, and when it is inserted into a cell, it may not necessarily replicate. It may not reproduce or it may not replicate. But when that fragment of DNA is inserted into the chromosome of the recipient cell, when it's inserted into the chromosome of the recipient cell, and when the recipient chromosome replicates, then so also that fragment of DNA which is inserted in the chromosome of the recipient cell will replicate along with the chromosome of the host or the recipient cell. So in that way, the DNA fragments, the number can be increased, or we can get identical copies of that fragment of DNA. Or we also have discussed that during the process of during the gene expression, that all the characters of an individual are determined by the process by protein synthesis, or by synthesizing a specific protein, and that information to synthesize protein is present in the DNA. So when that particular fragment of DNA which has been inserted in the bacterial or in the recipient chromosome codes for a particular protein, so when the objective is to obtain the protein of protein, then that DNA fragment can be allowed to express itself, can be allowed to synthesize protein, and as such protein can be obtained from that fragment of DNA. So that is also known as recombinant DNA technology. It involves the combination of joining two molecules of DNA from different sources, from the donor and the recipient, or from the donor DNA as well as the vector DNA. It's inserted into the DNA of another organism, so resulting into a new molecule of DNA called as the recombinant DNA, because it involves the combination of two molecules of DNA from different sources. Okay, so as I've said, recombinant DNA technology involves the formation of recombinant DNA molecule from two different sources. So this process of formation of DNA molecules, rDNA molecules, it does not take place independently, but it involves a series of tools, or it involves a series of enzymes. Then it has to be processed. It has to be subjected to other process. So let's see some of the tools that are used for this recombinant DNA technology. So the first is the enzymes. Okay, so these enzymes we know that these enzymes are biological catalysts, and they are specialized proteins. Okay, so these enzymes are of two types, basically exo and endonuclease enzymes. So as the name implies, these enzymes are the tools that cuts the DNA. So since the objective is to join the DNA of two molecules of DNA from differences, it has to be cut and then it has to be joined. So this cutting of the DNA is done by the enzymes. Particularly these exonuclease enzymes, they cut the DNA molecules at the 5' or 3' and at the ends of the DNA molecule. We know that DNA has polarity 5' end and 3' end. So this exo means exterior. So it cuts the DNA molecules at the 5' end or 3' end. And this endonuclease enzymes, these are the group of enzymes which gives internal cut and which cuts the DNA molecule within and not at the end. It gives internal cut. As such, these endonuclease enzymes, they are also known as molecular scissors. So these endonuclease enzymes, they cut the DNA within the molecule, not at the ends, and they are also known as molecular scissors. And there is a particular type of endonuclease enzyme known as restriction endonuclease enzyme. So this is a special type of enzyme where this enzyme is used for cutting the DNA, the foreign DNA or the donor DNA as well as the DNA of the vector. So these restriction enzymes, they recognize specific segment or specific nucleotide sequences on the DNA. So they are known as recognition sites. Restriction endonuclease enzyme recognizes specific nucleotide sequences on the DNA molecule and it cuts the DNA molecule at that site. So those special sites, they are known as recognition sites, and these recognition sites are special in a sense that the nucleotides are palindromic. Palindromic consists of a group of letters which reads the same forward and backward. So these palindromic sites or recognition sites have nucleotide bases which reads the same forward, which reads the same from 5' to 3'. So these are known as recognition sites and they have palindromic sequences. So for example, C, C, C and G, G, G. Okay, so when it's read from 5' towards 5', the same thing, it's the same word. So this is an example of palindromic sequence. So recognition sites recognizes the DNA molecules at the restriction endonuclease enzymes recognition the DNA molecule at the restriction sites and cuts the DNA molecule at that site. So as it cuts the DNA molecule at the restriction site, it produces ends. As it cuts, it produces ends, and so the ends may be blunt. It cuts the two strands of the DNA. DNA is a double-stranded molecule, so restriction endonuclease cuts the two strands and may produce a blunt end, or it may produce sticky end. So blunt ends are produced when restriction endonuclease cuts the DNA at specific position of nucleotide. So so that it produces blunt end. While in case of the other cut, called as the sticky end cut or cohesive end, restriction endonuclease enzyme cuts the DNA at a position slightly away from the center. So it produces single-stranded hanging ends, and these single-stranded hanging ends are known as sticky ends or cohesive ends. So this is how restriction endonuclease cuts the foreign DNA, as well as the same restriction endonuclease is used to cut the vector DNA. So this foreign DNA is inserted into the vector DNA, and this vector DNA, they are also known as vehicles or transporter vehicles. So they carry that foreign DNA into another organism called the host. So the same restriction endonuclease is used to cut the foreign DNA as well as the vector DNA. So this foreign DNA, it can be the DNA fragment of any organism. It can be the fragment of a microbe, DNA fragment of microbe or plant cell or animal cell. And these vector DNA, they are usually, mostly the most widely used vectors are plasmids. And you know, these plasmids, as we have discussed in this prokaryotic cell, these plasmids are extrachromosomal DNA. They are present in bacterial cell and they are extrachromosomal DNA capable of independent replication. So besides the bacterial chromosome, there is another chromosome which is known as the plasmid. So the same restriction endonuclease is used to cut the foreign DNA and the vector or the plasmid DNA, so that it produces the same sticky end, and these same sticky ends, they can form bonding during the formation of recombinant DNA. Okay, so that is one of the tool is these enzymes. So the other enzymes involve DNA polymerase, then even lysine enzymes, and then it involves even the use of lysine enzymes like lysine enzymes to degrade the bacterial cell, and the DNA polymerase to synthesize complementary DNA strand. So one tool is the enzyme. The other tool is the ligase enzyme, which is used for joining the fragments of DNA, and the other is the vector. So as we have said, these vectors are DNA molecule which is capable of independent replication, into which the foreign DNA is inserted inserted to be introduced into a host for replication or for cloning or to obtain a protein product. Okay, so these vectors, they have certain characters. They should have the vectors should have certain characters so that it is capable of transmitting or transferring that character to transferring the fragment to the host cell. So some of these characters are origin of replication. So vector should have an origin of replication, an oricide, and this oricide contains certain nucleotide bases where replication of the DNA is initiated, as we have already discussed with the replication of DNA. This oricide or origin of replication site where the replication of DNA inside the host cell is initiated, and then it should also have selectable markers. The vectors should have certain selectable markers with which it imparts the cell. Okay, selectable markers by means of which the cells can be identified. For example, a bacterial plasmid with a selectable marker, when it is again reinserted into a bacteria, that selectable marker enables the bacteria to be identified from the bacterial cells which do not have the plasmid. And many of these selectable markers are resistant gene, resistant to antibiotics like ampicillin, tetracycline, chloramphenicol, terramycin, etc. So these resistant genes, they are used as selectable markers and enables the bacteria the transformed bacteria to be identified or to be isolated from the non-transformed bacteria, selectable markers. Okay, and then they also, vectors should also have a cloning site. They also should have a cloning site and or recognition site. So this cloning site or recognition site are the sites having specific nucleotide sequences where the restriction endonuclease recognizes it and cuts the vector DNA at that site. And after cutting the DNA at that site, then the foreign DNA is inserted into it. That restriction endonuclease may cut the foreign the DNA, the vector DNA, at the recognition site or a little further away from that. So it should have at least 1 or 2 recognition sites by means of which the restriction endonuclease enzymes can recognize it. Okay, then these vectors should also be small in size. They should be small in size and they should have low molecular weight, because the larger molecules, they collapse during handling of it, or it's difficult to manipulate larger molecules. So vectors should be small in size and they should have low molecular weight. And the other tool is host organism. So the host organism is the organism into which the recombinant DNA is to be inserted. So it could be even a bacteria, it could be plant cell or animal cell, and the other tool is the foreign DNA. So the foreign DNA, it can be a complementary DNA or a synthetic DNA, synthesized DNA from the lab or in vitro, or complementary DNA which is synthesized from the messenger RNA. So these are certain tools, and with this tool, with the knowledge of these tools, we will proceed with the steps of recombinant DNA technology in the next class again. So we'll discuss with the steps, and I hope to see you again.